Hi everyone, I am sorry I couldn't join live, but I do appreciate the invitation and I'm excited to share with you some of our global perspectives and research on human milk and infant microbiomes. So here are my disclosures. And I wanted to begin by thinking about infant microbiomes and how they are different or the same around the world. So this is research by Shelley McGuire and her team at the INSPIRE study, and you can see here that they very meticulously collected infant stool samples from infants in 10 countries around the world. And you can see here that their microbiome profiles are quite different, so there's variation geographically. And an interesting question is what drives these differences in different parts of the world? More recently, this beautiful study came out in Nature Communications, where again, they looked at infant microbiomes from countries around the world. And here, of course, there are differences, but they're highlighting common patterns of development around the world. And so we could also ask, what drives these similarities in infants' microbiomes around the world? And so I would say the answer to both of those questions, what drives similarities and differences in infant gut microbiomes around the world, is breast milk. So this is a beautiful study from the Teddy study led by Chris Stewart, and they showed that very clearly breast milk is the biggest factor shaping infant microbiomes in early life. So here you're looking at 3 different time periods, 3 to 6 months, 7 to 10, 11 to 14, and a variety of different factors that were tested for their ability to explain variation in the infant microbiome. And very clearly, especially early in life, what you can see is that breast milk is the biggest one. And so, of course, a question is why? And how? Why is it that breast milk is shaping the microbiome and how does that work? And so here I think we need to think about what's in human milk and what it does. So human milk, of course, is a food. I would argue it's the original superfood. So it's got all the nutrients a baby needs to grow and develop early in life. But it's more than that. We also can think of milk as a baby's first vaccine. So it contains immune factors, including maternal antibodies, that help protect babies from pathogenic infections. These antibodies also actually bind to commensal good microbes and help them colonize the infant gut. We could think of milk as a personalized medicine. So it's personalized because it's different in each mom and it's reacting to signals from the baby that are passed back to the mother during the act of breastfeeding. It's also dynamic. So milk is changing over time. As the baby grows day to day, month to month, its needs are changing and the milk is changing along with it to match those changing needs. And then, of course, human milk is tailor-made for tiny humans. So all mammals make milk. This is a defining characteristic of mammals. But I put 3 examples here. If you think of a baby human compared to a baby cow or a baby pig, they're very different. A baby cow is up wobbling around almost right away, so it needs strong muscles, whereas a baby human is sleeping for a lot of its first life, and it's not up walking around for many months. Instead, it's growing its brain. So you can imagine the composition of milk for a cow or a pig or a human is going to be very different. And more recently, we're learning it's not just that milk is different for every species because of that infant or baby. It's also made for the baby's microbes. So the microbiome of a baby human versus a baby pig versus a baby cow is also different. And milk has evolved along with these microbial communities to optimally feed microbiomes. This is a bit of an outdated by now article, but it's still a great one, and I encourage you to check it out if you're interested in a more lay perspective on breastfeeding and the microbiome. So how is it that breastfeeding shapes the microbiome? Well, we have to think about what's in the milk. So these are 2 pictures telling you the same story, which is that even if you can't read all these tiny words, there are many things in breast milk that simply are not found in formula, the alternative. And here on the left, you can see some of those major compounds. So formula and breast milk both contain the nutrients that a baby needs to grow and develop. But on top of that, milk contains many non-nutrient components. So we talked about antibodies, there are also hormones, growth factors, and so on. All of these are important for infant development, and many are also important for the microbiome. And I would say this figure actually needs an update because it doesn't contain 2 very important factors for the microbiome. So one of those being probiotic or live beneficial bacteria, and another being prebiotic oligosaccharides, which are food for those beneficial bacteria. So both are naturally contained in human milk. And if you're saying, but Megan, formulas these days also have probiotics and prebiotics. That's true, but I have to point out that it's not the same. It's not at all the same. So human milk naturally contains hundreds of strains of bacteria and hundreds of different oligosaccharides. But in formula, they're adding a couple. And so that's good. It's getting closer to human milk, but it's not at all the same and it's not personalized or dynamic in the way that human milk naturally is. So shifting to tell you about some of my own research, a lot of this has been done in the CHILD cohort study. CHILD is a pregnancy cohort of over 3,500 families recruited back around 2010. We're still following these babies who are actually now teenagers. And this study was formed to really understand what is it about this generation of children that's causing an increase in allergies and asthma compared to prior generations. And to do that, the researchers involved, which include people from over 20 disciplines, so a really big team, thought, well, we need to capture the environment because that's what's changing. Genetics aren't changing, but the environment is changing. The environment includes the indoor environment, the outdoor environment, things like stress and nutrition and weather, and of course, the microbiome. So this actually wasn't part of the initial child study design, but shortly after it got started, around 2010, is when the microbiome became a big deal. And fortunately, the study had collected stool samples. So this is where I first got involved as a postdoc, studying the gut microbiome of these babies. A few years later, by the time I was starting my own lab, I realized they had also thought to collect breast milk samples, and nobody was yet interested in those samples. So by now we had caught on that breastfeeding was really important to the microbiome, and I decided to take hold of those samples and see if I could understand what it is about the breast milk that's shaping the microbiome. And so this is a linear view of the study. You can see, as I mentioned, recruitment during pregnancy back in 2009. There have been follow-ups all throughout into teenagehood, microbiome collection at several different time points. What I'll focus on today is data from the first year of life. So stool samples were collected, breast milk was collected at one time point, and these swabs represent nasal samples, which we've also been able to use to characterize the microbiome. So focusing in for a moment on the milk, this is the same figure you saw earlier, but updated with those pre and probiotics. And so what you'll see is that over time, we've been able to chip away at this rainbow of milk components and analyze nearly all of them, which is really exciting because we have the opportunity now to look at how milk as a biological system of all these many different components is functioning as a system to influence the microbiome and development. And so many of these data sets were published individually over time. Some of them are still coming soon. But what I'll tell you today is just one example of a story from the child study using milk data along with microbiomes. So this was a collaboration with Liat Shenev at NYU, as well as PJ Subro, the director of the child study, and Mike Surrett, who led the microbiome analysis for the nasal samples, and one of my fantastic analysts, Kelsey Fair. And so what you can see here is that our overall finding in this study was that the microbiome develops both in the nose and in the gut at a gradual pace, normally. And it's breast milk that's driving this development. So in babies who were exclusively fed human milk until at least 3 months, we saw this gradual development. But in babies who weaned early and started consuming formula, the acceleration was premature. And this premature acceleration was associated with a higher risk of asthma later when the children were 3 to 5 years of age. And so how does this work? Well, once again, we found, just like the Teddy study, that it was really breastfeeding among all these other potential factors that it was explaining variation in the nasal microbiome and the gut microbiome of these children. We further were able to show that breastfed infants had a lower risk of asthma, and it really was these microbial communities, both in the nose and the gut, that were mediating this impact of breastfeeding on protection from asthma. We're able to go one step further and identify specific microbes and specific milk components that were driving this relationship. So on the left here, you can see that milk components on their own, the orange line, were somewhat predictive of asthma, and gut microbes and nasal microbes on their own were also predictive, but it's really when you put everything together that you get this best ability to predict which infants will go on to develop asthma. And then on the right, we were able to look at feature importance scores. So specific milk components and specific microbes are shown here in order of importance in terms of which ones helped us predict the infants that would go on to develop asthma. So for me, the motto of this story is timing is everything. A healthy infant microbiome is not just about having the right microbes, it's about having them arrive in the right order at the right time. So there were microbes that seemed to be good, but that's only if they appeared early, and vice versa, microbes that seemed to be good, but only if they appeared for the first time later in infancy. And it's really exclusive breastfeeding in the first months of life and the dynamic nature of human milk that regulates this intricately structured microbiome development. So I like to sometimes think about this as a dessert. This is an Nanaimo bar. It's popular in Canada, but I know we have a global audience, so tiramisu maybe is a better example. But either way, the point is that these beautiful desserts, which you could think of as a healthy baby microbiome, require ingredients, but you cannot just throw all the ingredients in the bowl at once and hope to have this beautiful layered dessert come out. You need to add the right ingredients, but also in the right order at the right time. All right, so shifting to global perspectives, everything I've told you so far from our research is about the Canadian child cohort study, but I'm now leading an international consortium of breast milk researchers in 4 countries around the world, including 3 low middle income countries that you can see here. And our question really is, what are the determinants and consequences of global and individual variability in human milk composition? So these, as you can imagine, are 4 very different contexts geographically. Altogether, we have over 1,000 mother-infant dyads participating in the study, and her goal really is, again, to analyze that whole rainbow of milk components and then understand with some sophisticated data science approaches, how do all these different milk components relate to infant health outcomes, including growth and others as well. So the protocol for this study was published recently. I do want to really encourage secondary analysis of this data. So if you're interested in tapping into the IMIC data, please reach out and let me know. We're asking many different questions, of course, with this data. We're writing up the main papers as I speak, so stay tuned for those. I will just give you a glimpse of the answers to that first question, which is, what are the ranges and distributions of analytes in human milk in different geographic settings? So here, what you're seeing is each dot is 1 milk sample, and each color is a different study. So in green here, we've got the MISAM study in Burkina Faso, and there's 3 shades of green because 3 time points were analyzed. In red, you have the child cohort, which I already talked to you about, only 1 time point analyzed. So what you can see here is the conglomeration of over 11,000 components that we measured in each milk sample. And you can see that the samples from each study tend to cluster together, indicating that samples from different studies or different areas of the world are different in composition. Maybe that's not surprising, but I think it's pretty fun to see in a picture. What you also can see is that there's a temporal pattern. So as I mentioned, milk is changing over time. And you can see here the green shaded dots are earlier in infancy and the darker purple ones are later in infancy. So we see both differences by setting and differences by time of lactation. And so there's a lot more to come from IMIC, but I'll give you one more example specific to the microbiome since that's our focus today. So led by Melissa Manis and Kelsey from my lab, Melissa now has her own lab at the University of Texas. We did a deep dive into the microbiome of these samples in 3 of the studies. And what you can see is that there is differences in the microbiome in different countries, but there's also a shared sort of core milk microbiome of 23 taxa that we found in all 3 studies. So again, this idea that yes, there are differences, there are also similarities in human milk around the world. This was also shown in the INSPIRE study that I mentioned earlier by Shelley McGuire's team. So I showed you the gut microbiome from that study earlier. This is the milk microbiome. Again, similarities, but also differences around the world. So in summary, I hope I've been able to highlight for you that infant microbiomes vary geographically, but also follow some consistent developmental patterns globally. If we think of human milk, it also compositionally varies geographically, but still follows consistent temporal patterns globally. I showed you that human milk is the most important driver of early microbiome development, both in the gut and in the nose, and that it's acting as a pacemaker of microbiome development with long-term health implications, with one example being asthma that I showed you today. And finally, I hope all of this will make clear to you that human milk really offers inspiration for microbiome-targeted approaches to health promotion and disease prevention. So if we can learn from human milk which microbes or which prebiotics or which immune components are useful for driving healthy microbiome development, that's knowledge that we could use to develop microbiome-targeted approaches to preventing disease or promoting health in infants for sure, and maybe even across the lifespan. So a couple fun things before I sign off. If you check out our website, you'll find we do fun things like videos and podcasts and AMAs on Reddit. And this is all to share the knowledge about human milk and the microbiome, and you're welcome to check it out and share with your friends. We also have a milk club. We meet every month together in Manitoba, but 3 or 4 times a year, we open it up to people globally. So if you'd like to join us for those types of meetings, then this is where you can find me. I, of course, want to thank everybody involved, both in my lab, in the child study, the Milk Center, and of course the IMIC Consortium. And with that, I will say thank you very much. I'm sorry I'm not there live to answer questions, but please do reach out if you have questions. This is where you can find me.